So where are we headed today? We're going to pick up this idea of hormone actions and the hypothalamic pituitary axis, and I'll remind you that what we talk about today will be covered in the exam on Wednesday. So where we're headed is we're going to talk first about the hormone effects, and we'll do this very briefly because we covered this last time. Just remember that the hormone effects are to increase enzymatic activity, increase channel activity, to increase endo and exocytosis, and affect gene transcription and translation. So hold on to those ideas. What we're going to do today is we're going to really talk in some detail about how this will happen. And we'll talk first about the hormone receptor complex, and the first one we're going to talk about is the hormone receptor complex that we initially talked about last time, but we're going to drill down today and now start talking about a particular hormone receptor complex formation. We're going to talk about the large hydrophilic hormones. These are the ones of the peptide hormones or protein hormones. And we're going to see that many of these will work through something called a G protein coupled receptor. We're going to tell the story of the large hydrophilic hormones through this G protein coupled receptor. If you take an endocrine class, you'll find that there are a whole host of other forms of receptors, particularly the receptor tyrosine kinase receptors. You'll see those in the endocrine system, you'll see them in development, you'll see them all over the place. But we're going to fundamentally stress this one called the G protein coupled receptors. Something like 4% of the genome is taken up by genes involved with generating these G protein coupled receptors. All of these proteins have a common configuration. And that common configuration is that they traverse the membrane 7 times. They're said to be 7-pass transmembrane proteins. What else they also have in common is that all of these G protein coupled receptors are activated by something known as a Gtp binding protein. We'll see in this story that there's something called the trimeric, heterotrimeric Gtp binding protein. That's a fancy term meaning that these Gtp binding proteins are made of 3 subunits, an alpha, a beta, and a gamma. We're going to emphasize mostly the alpha subunit. The alpha subunits then can come in several major types. The first one we'll talk about is G alpha S, and others G alpha I. We'll run into this. We talk about the cardiovascular system, and to some extent GQ. Now, this is an enormous group of proteins. But what I want to emphasize is how these proteins bind a hormone. Once they bind a hormone, that hormone now communicates through this transmembrane protein across the phospholipid bilayer and communicates to the cytoplasmic side of the cell. That communication now involves the binding of the alpha subunit of the so-called trimeric Gtp binding protein. The alpha subunit, when it has Gtp bound to it, and we'll cover this in some detail, when the alpha subunit has Gtp bound to it, it activates something called adenylyl cyclase. But the end result of the adenylyl cyclase is to upregulate kinases. Another term here that's going to be important, we'll see over and over again, these kinases, this means protein phosphorylation. Anytime you phosphorylate a protein, you can change its function, and we'll talk a little bit about that. And in contrast, then we'll talk about the hydrophobic hormones, where the hydrophilic hormones are communicating to the cell across the phospholipid bilayer through these transmembrane proteins. We'll see that the hydrophobic hormones, which include thyroid hormone and the steroid hormones, have the capacity now to bind to cytoplasmic receptors. Because of their hydrophobic nature, because of their so-called lipophilic nature, they easily pass through the phospholipid bilayer and access the cytoplasm. Once inside the cytoplasm, they can bind to cytoplasmic receptors. And these cytoplasmic receptors now can enter the nucleus and function as a transcription factor. So what we'll see then is that we'll bind the hormone to a particular region of the cytoplasmic receptor in the cytoplasm. That will trigger an access through the nuclear pores. There will then be a DNA binding domain, and that will begin the process of activating the gene by beginning to recruit other proteins to function as a transcription factor. So we blithely talked about transcription factors earlier on, but here we're going to see that now these hydrophobic hormones, particularly the steroid hormones, can affect gene transcription and translation by binding these cytoplasmic receptors. Then the last piece is going to be the hypothalamic pituitary axis. This is a really important aspect of all hormonal regulation. And there are a few main ideas I want you to carry away from when we talk about the hypothalamic pituitary axis. First, we'll talk about the posterior pituitary. The posterior pituitary is nothing more than an extension of the hypothalamus, and basically there are neurons in the hypothalamus that send their processes into the posterior pituitary. There are only two major hormones that are released from the posterior pituitary. This is oxytocin. Oxytocin is important in causing uterine contractions. It's also important in causing smooth muscle contraction for the release of milk. The one that we'll see later on in lectures on the kidney is vasopressin. Vasopressin is regulating water and salt balance in the kidney. We'll talk a little bit about that later on. But these two I'll ask you to hold on to. But the major idea here is going to be to contrast the structure of the posterior pituitary and the anterior pituitary. The posterior pituitary is an extension of the hypothalamus, but the anterior pituitary originates in another way. The anterior pituitary originates as epithelial cells in the pharynx during development. And there are a group of these epithelial cells which then bud off. They break off from the pharynx and they migrate as a group just underneath the hypothalamus. Now, I say that not to give you the developmental story, but rather to emphasize that these epithelial cells now are going to be separate and distinct from the hypothalamus. They never get into the brain. They're always separate from the hypothalamus. The only communication between the hypothalamus and these cells in what will develop into the anterior pituitary is through something called the hypothalamic pituitary portal system. So blood will flow through the hypothalamus, and then subsequently it will flow from the hypothalamus into the anterior pituitary. So as the blood flows through the hypothalamus, there will be certain neurons in the hypothalamus that will secrete releasing hormones. The releasing hormones which come out of the hypothalamus will be carried then to the anterior pituitary in the portal blood. Then in the anterior pituitary, the releasing hormones bind to troph cells. Troph cells then release stimulating hormone. The stimulating hormone then activates the gland, such as the thyroid gland or the ovaries or the testes, and then those glands release their hormones. Those hormones then feed back onto both the hypothalamus and onto the anterior pituitary in something called negative feedback. We'll try to get through all of that. But before we get too deep into it, what I want to remind you is where we ended up was the discussion of the formation of the hormone receptor complex. The first one we're going to talk about is the binding of hydrophilic hormones to transmembrane protein receptors. So again, remember when we think about the phospholipid bilayer, where this is outside and this is inside, that those large hydrophilic hormones, whether they're peptides or proteins, did not have ready access across the phospholipid bilayer. So the signaling across the phospholipid bilayer is through a transmembrane protein. I'm going to simplify this a lot. It comes across the membrane 1, 2, 3, 4, 5, 6, 7. So this is a transmembrane protein. This is said to be a 7-pass transmembrane protein. The 7-pass transmembrane protein now communicates from the outside to the inside. Now, I've simplified this a lot by just putting these squiggles across the membrane. Each of these squiggles is meant to represent an alpha helix. But in the extracellular region and in the cytoplasmic region, these cytoplasmic tails will form a 3-dimensional structure, and these extracellular regions will form a 3-dimensional structure that, in contrast, the simple U-shaped thing that I'm showing as a receptor provides the receptor. But remember, over and over again, I can't emphasize this too much, the hormone function is determined by the receptor. To make this active, the receptor determines the hormone function. Now, of course, from last time, you'll recognize that we have to have the proper binding, the 3-dimensional fit between the hormone and its receptor. Now, in ways that still aren't quite clear, the binding of the hormone in the extracellular region changes the structure of the cytoplasmic region of this protein. So the hormone binding affects the 3D structure of the cytoplasmic portion of the protein. Well, across the membrane, actually embedded in the membrane, these proteins are associated with the membrane, but that's a detail. There'll be an alpha and beta and a gamma subunit. So this is the alpha, the beta, and the gamma. This is said to be a hetero trimeric Gtp binding protein. Well, it's trimeric because there's an alpha, a beta, and a gamma subunit. It's a Gtp binding protein because this alpha subunit has the capacity of binding a molecule of Gtp. Now, I should say, and should emphasize at this point, that when the alpha, beta, and gamma are all living together, when these guys are all clustered together as the trimeric subunit, of the 3 subunits rather, the alpha is bound to guanosine diphosphate. Just as a word here, remember that adenine is a purine. There's a related purine called guanine, where we can make ATP. We can similarly make guanosine triphosphate. So this is guanosine triphosphate. It can be dephosphorylated to guanosine diphosphate. Well, when the alpha and beta and gamma subunits are all together, hanging out with their tails as part of the phospholipid bilayer, these usually contain fatty acids which lash them into the phospholipid bilayer. When they're all hanging out like this alpha subunit is inactive. So under these circumstances, this alpha subunit doesn't do anything. However, when this threesome comes up against the G protein coupled receptor, this G protein coupled receptor with its hormone bound has now undergone a conformational change which will allow the alpha subunit to bind to this protein. So you can imagine this purple alpha subunit sidling in here with its GDP attached. The binding of the alpha subunit to the activated hormone, the hormone that has been bound, the alpha subunit with the GDP attached, bound to the transmembrane protein, the G protein coupled receptor with the hormone attached, allows down here for an exchange. The GDP leaves, and it's exchanged now for Gtp. This is therefore a Gtp binding protein. It binds Gtp. The alpha subunit is the only one of the three subunits that binds Gtp. The most important aspect of this is where the alpha, beta, gamma subunits, when living together with the GDP attached, are not active. The replacement, the exchange of this GDP for Gtp, and that's exactly what happens. The GDP leaves and is replaced by a Gtp. This now becomes active. So let's spell that out. Let's take this panel and describe this as the activated function of the activated alpha subunit. Now, the specific subunit we're going to talk about here is called G alpha S, and S stands for stimulatory. A little later, we'll find that there's another variant of this called inhibitory, but let's keep this a little simpler for today. This is our box, it's the cell. Across the membrane, as we showed above, is this 7-pass G protein coupled receptor. As we described above, it had bound here its hormone. That had given it a conformational change in the cytoplasmic side. The alpha subunit had now bound to the cytoplasmic side of this. Let me make this a little simpler. This alpha subunit has bound to this and exchanged its GDP for Gtp. So what we've drawn above, we now have here. The beta gamma go off and do something else. And if you get into an endocrine course, you'll see the beta gammas do a whole flock of things. We're not going to get into that here. What I want to emphasize is what the alpha subunit does. The alpha subunit, the activated alpha subunit binds to a protein called adenylyl, Adenylyl cyclase. Now, there's meaning in this term, adenylyl cyclase. What the adenylyl cyclase does is the adenylyl cyclase converts adenosine triphosphate, ATP, into cyclic AMP. I'll show you a picture of this later, but basically what it's going to do, it's going to take this number 5 and number 3 carbon of ribose. So this has an adenine out here. This is 1, 2, 3, 4, and this is the number 5 carbon. This 5 carbon has the OP, OP, OP. That's a triphosphate. Won't go into all those details. But what adenylyl cyclase does is it takes this hydroxyl down here and forms a new bond that goes O, P, O. So it basically, this is the adenylyl cyclase. The adenylyl cyclase forms something called CAMP. Not surprisingly, often referred to as CAMP. All it's doing is an enzyme that's now taking the number 3 carbon and its hydroxyl and forming this phosphodiester bond to the number 5 carbon. So this is a 3 to 5 phosphodiester linkage. Well, I won't go through all of that here. I'm just going to say that we're going to take ATP, and through its activation of this protein that actually crosses the membrane 12 times, this is Adenylyl Cyclase. Adenylyl cyclase takes this ATP and converts it into cyclic AMP. Now, cyclic AMP is what's known as a second messenger, because the second messenger here, the cyclic AMP, has the capacity to come out here to this green protein, which is a cytoplasmic protein, called protein kinase A. Now, if we get down into the weeds of this, you're going to find there are protein kinase A's, there are protein kinase C's, there are a number of them. But this is the one that's turned on by cyclic AMP. What I want you to focus on, though, is whenever you see this term kinase, think an enzyme that phosphorylates proteins. Now, it turns out that the only amino acids that it adds a phosphate group to are serines and threonines. Now, the only reason I bring this up is these are serine, threonine kinases. The reason you can phosphorylate serines and threonines is they have a hydroxyl on them, and you'll use that hydroxyl to add the phosphate. But that's a detail. What I really want you to recognize is that the protein kinase A is an enzyme that adds phosphates to proteins. And anytime you phosphorylate a protein, down here, protein phosphorylation changes its function. So that when you modify a protein by adding these great bulky and charged phosphate groups, you change its function. One of the places we see that commonly is if there is a transmembrane protein here, which is a channel. And if we modify this with a PO4 with a phosphate, we phosphorylate it. If we phosphorylate this channel, we change its opening and closing properties. So there's a classic example of how these enzymes, well, there's a classic example how we use a G protein coupled receptor to activate an enzyme, adenylyl cyclase, that in turn affects the probability of a channel opening and closing. Because remember, we said last time that the kinds of things we can affect with hormone regulation are we can affect enzyme activity, adenylyl cyclase. We can affect channel opening and closing. There's a transmembrane protein being a channel. We can affect exocytosis and endocytosis. These also appear to be through phosphorylation events. And finally, we can affect gene transcription and translation. We're not going to emphasize it here, but if you take another cell biology course, you'll learn about something called Krebs and Krebs binding protein. And what that is a means of using cyclic AMP to affect gene transcription and translation. We're going to leave that out. For our purposes here, we're going to divide the world into these two great categories. We're going to talk about the hydrophilic hormones communicating to the cell through these transmembrane proteins by upregulating an enzyme called Adenylyl Cyclase that generates cyclic AMP that now turns on a kinase to phosphorylate enzymes. If you hold on to that cascade of events, that's all I'll ask. If you take another cell biology course, you'll find there are many variations on this theme. So any questions to hear? Yes?
I guess I'm a little bit lost here on how the alpha, beta, and gamma subunits kind of relate to these concepts.
Let me show you a slide. Let's see if we can straighten this out a little bit from that, and then I'll take that question again. So this is the structure of the G protein coupled receptor. It grows across the membrane 7 times. So this is basically just what I drew on the board. The 7 transmembrane protein regions, the N terminals on the outside, all of these extracellular loops will form a 3-dimensional structure to which the hormone can bind. Down on the cytoplasmic side, we're going to have the region that can interact with the Gtp binding protein. So what will happen is the binding of the hormone on the outside changes the shape. It can be down in here. It's probably through all of the 3-dimensional shapes generated by those cytoplasmic regions. Then this is stylistic drawing of what I just put on the board. So let's try it again. So here's the 7-pass transmembrane protein receptor. We've glibly put in the alpha, beta, and gamma subunits. Often these are shown as being connected to the cytoplasmic leaflet because they contain fatty acids which tie them in to the phospholipid bilayer, but don't worry about that. We have the alpha, beta, and gamma subunits here. The important player for our purposes today is the alpha subunit. Normally, when these 3 are all living together as a happy family, alpha, beta, gamma, the alpha has a GTP attached to it. But then when this threesome binds to an activated G protein coupled receptor, in other words, a G protein coupled to the receptor that's bound to hormone, whose cytoplasmic region has had its conformation changed, then the alpha, beta, and gamma bind to this site. That confers a change such that the GDP is lost and is replaced with a GTP. It's actually an enzyme here that transfers it. You basically clip off this GDP and add a GTP. Now, with this alpha subunit and its GTP attached, that now allows the beta and gamma to come loose. They go off and do something else. Turns out they'll open channels. We're not going to deal with that. They go off and talk to somebody else. The one we're interested in is the activated alpha subunit with this GTP attached. What it turns on is adenylyl cyclase. This adenylyl cyclase is not quite accurate here. This adenylyl cyclase crosses the membrane like 12 times, so it's an integral membrane protein. But what the adenylyl cyclase does now is it converts ATP to cyclic AMP. The cyclic AMP is now the important actor. The cyclic AMP now activates protein kinase A, and this goes out and phosphorylates a whole bevy of different proteins. And so this is a cascade of events. The cascade of events is you bind the hormone, you activate the trimeric Gtp binding protein, you activate the adenylyl cyclase, and then end up phosphorylating a whole bunch of other proteins. Now, one of the major things to hold on to here is the idea of amplification. That, remember, hormones commonly function at very, very low concentrations. You don't have a lot of them out there. So in this case, you can bind one hormone molecule to this receptor and perhaps turn on maybe 10 or 100 of these alpha subunits that then go out and turn on another 10 or 100 adenylyl cyclases, which then in turn produce maybe 10 or 100 cyclic A's. And then by the time you get out here, you've amplified the initial binding of the hormone maybe 1,000-fold. So you bind one hormone molecule and end up phosphorylating 100 or 1,000 proteins. So you get this tremendous amplification effect through this cascade. So does that answer your questions? Okay. Then this is simply what I drew on the board. And it's showing the adenosine triphosphate. So here's the adenine out here on the number 1 carbon. Here is the triphosphate. The adenylyl cyclase now forms a new phosphodiester bond. It uses the energy in this triphosphate to form this number 3 to number 5 bond. So now we have an oxygen, phosphorus, oxygen. That's the 3 to 5 phosphodiester linkage. And here we have the cyclic AMP. And it can be now inactivated by a phosphodiesterase, breaking that bond. And there was a question in the back. So the question is, great question, how long does it take between the binding of the hormone and getting this cascade? Seconds to minutes. When we get into the nervous system, we'll find that binding a neurotransmitter to a receptor will open a channel. That's a millisecond time scale. This is a seconds to minutes time scale. If you start getting in gene transcription and translation, then you're talking about hours to days kinds of time scales. So you have this tremendous variation from binding a neurotransmitter to a receptor to get a channel to open in milliseconds up to times of hours to days with gene transcription and translation. And then, yes? So the question here is, are you telling me that the alpha, beta, gamma, this trimeric Gtp binding protein will bind to the cytoplasmic side of the hormone receptor so they can come apart? That's exactly why it is. Well, I should be a little more careful. It's not just so they come apart. It's so that the exchange factor can take the GDP off of the alpha subunit and replace it with a GTP. So that's the major idea, is once these guys are all here, that activates an exchange factor that lops off the GDP and replaces it with GTP. And once it has a GTP attached, then it's an independent actor as an alpha subunit and goes out and does something. Over time, then, this GTP will be converted back to GDP. And when it's converted back to GDP, it will find its beta gamma subunit and then quietly sit there waiting to be activated again. Okay. Anything else? Yes.
So the alpha subunit that's attached to the transmembrane protein there, is that the same one that's attached to the
Adenylyl cyclase?
Yeah. Does it like move from one to the other, or are those two different alpha subunits?
So the question is, are we talking about the same alpha subunit, which is bound to the Gtp binding protein, goes out and activates the adenylyl cyclase? That'll be the same one. And one of the things that's not very well shown in this figure, but this alpha subunit will be attached to the phospholipid bilayer, so it's free to move back and forth in the plane of the membrane. So we can wander off and activate adenylyl cyclase, which is a transmembrane protein. So you remove the three-dimensional diffusion into a two-dimensional plane. Okay. We better move on. Well, that provides a story for how these hydrophilic hormones affect cell function. But now let's take a couple of minutes and talk about the other characters that we discussed last time on Friday, which are the hydrophobic hormones. And you'll remember we talked about two major hydrophobic hormones. We talked about thyroid hormone, and we talked about thyroid hormone, and we talked about the steroid hormones. But what those two had in common is that they were hydrophobic, that they had to attach to a protein transporter, a carrier protein, and that even though that affinity between the transport protein, the carrier protein, and the hormone was loose, it would come and go. And where we left off was to have the phospholipid bilayer. And imagine one of these cholesterol-derived steroid hormones. We could pretty much tell the same story for thyroid hormone. The idea is they're hydrophobic. These ring structures don't have much charge on them, and so they go zipping through the membrane. So now these hormones are in the cytoplasm. But the rule still is the hormone must bind to a receptor. There has to be specific binding between the hormone and its appropriate receptor. It's just in this case, for these steroid hormones and also for the thyroid hormones, the receptor is a cytoplasmic receptor. In contrast to the transmembrane protein we have above, this is a cytoplasmic receptor. Now, I'm going to simplify this a whole bunch. And this structure here is meant to be a steroid hormone receptor. Now, the thyroid hormone receptors are a little different because they're in the nucleus, but for our purposes, we're going to summarize them as being the same. And we're going to say that out here, at this end of the molecule, is the hormone binding domain. Turns out that these molecules are pretty linear, such that this is the amino terminus and this is the carboxy terminus. But this region in here in black, that's where the hormone will bind. So the hormone will come into the cell and bind to this site. The binding of the hormone to this region of the receptor activates a region here called the nuclear access domain. Now, after all, you'll remember from last time that these steroid hormones are classically serving as transcription factors. And as transcription factors, they have to get into the nucleus. If they're going to be a transcription factor, remember, all transcription comes from DNA that has to be nuclear. So the nuclear access domain is turned on by the binding of the steroid hormone. That allows this entire complex to enter the nucleus. Then in the nucleus, there's this next region of the molecule that's really important. This is called the DNA binding domain. So again, if you're going to be a transcription factor, you need to be able to bind to a promoter. You have to bind to the DNA. So the binding of the hormone to the receptor says, okay, let's go to the nucleus. That turns on the nuclear access domain that allows this molecule to be recognized by the nuclear pores. This now opens the portcullis, allowing the complex to get into the nucleus. Once inside the nucleus, there is this region of the protein, which is the DNA binding domain, and it's going to seek out a particular promoter and bind to a particular promoter. And when it binds that particular promoter, it's going to now activate this region out here called the transactivation domain. And what the transactivation domain does is it helps assemble the component proteins of the transcription factor. Remember, we said there were some 13 or 15 different proteins involved in a transcription factor. Well, this, in many cases, this hormone receptor with its hormone bound is part of that transcription factor complex. to turn on a gene. Now see coming together the importance of the promoter in the DNA and its interaction with these hormone receptors that are cytoplasmic receptors going into the nucleus to turn on genes directly. So any questions to there? So hold on to the idea now that up above we're using hydrophilic hormones to communicate across the phospholipid bilayer with that cascade of events. Here we're using a hormone that gets into the cell to turn on to activate a receptor that now acts as part of a transcription factor to turn on certain genes. Yes?
I'm sorry, can you further explain what you mean by assembling the proteins of the transcription factor?
Okay, so what do I mean by assembling the proteins of the transcription factor? A transcription factor isn't just one protein. It's a whole series of proteins. You can think about them as a series of Legos that have to come together to generate a certain three-dimensional shape to allow for the binding of the RNA polymerase. In this particular case, one of those important proteins is this receptor hormone complex that is bound to the DNA. And so then that assembles the further proteins that form the full transcription factor, which attracts or recruits the RNA polymerase to start making the RNA from that particular gene. Now, in the last few minutes, let me talk about the hypothalamic pituitary axis. The hypothalamic pituitary axis is going to include the hypothalamus, which is part of the brain, the anterior pituitary, and the posterior pituitary. We're going to draw the posterior pituitary and the hypothalamus first. This way. The hypothalamic pituitary axis. So there's an extension here of the hypothalamus. So this structure up here is the hypothalamus. And there are neurons in the hypothalamus that send their process from the hypothalamus out into the structure here, which is known as the posterior pituitary. These neurons, whose cell bodies are in the hypothalamus, will in fact secrete the hormones vasopressin, which are for kidney function, and oxytocin. And this is for smooth muscle contraction, particularly uterine smooth muscle contraction during parturition. The major idea here is the posterior pituitary is an extension of the hypothalamus. Now, in here in red is a structure, let me put it in orange. In here in orange is a structure known as the anterior pituitary. The anterior pituitary is secretory cells of epithelial origin. It's a place called Rathke's pouch. It's an outpocketing of the pharynx during development. It migrates up here, but there are a whole series of cells in here. These will be called troph cells. the only communication between the hypothalamus and the anterior pituitary is through a series of blood vessels. So there are going to be these blood vessels that flow through the hypothalamus and then through the anterior pituitary. This set of blood vessels is called the hypothalamic pituitary portal system. So where the posterior pituitary is an extension of the hypothalamus itself, the anterior pituitary is a separate set of cells and only communicates with the hypothalamus through the blood vascular system. So in the hypothalamus are these so-called neuroendocrine cells. That releasing hormones. The releasing hormones now enter the hypothalamic pituitary portal system. The releasing hormones have receptors on cells in here called troph cells. So the troph cells have receptors for releasing hormones. So they have receptors for releasing hormones. The releasing hormones, let me do it this way, that signal the troph cells to release stimulating hormones. The stimulating hormones then bind to receptors on target glands. And there's one more piece I have to put in here. I'm sorry, but I have to put it in. There's a 4-box model. Deep 6 this. And the 4-box model goes as follows. This is the hypothalamus. This is the anterior pituitary. In purple is the target gland. The target gland could be, say, the thyroid gland. So from what we see above, the releasing hormones, signal the anterior pituitary to secrete stimulating hormones. And the target gland now releases its hormone. Now in the final moment here, this is really important, this hormone released from the target gland will feed back onto the anterior pituitary and feed back onto the hypothalamus, and this will inhibit the hypothalamus and anterior pituitary. So, what happens if you remove the target gland? If you remove the target gland and you no longer have hormone, what's that going to do to the releasing hormones, the stimulating hormones? I'll leave you with that, but you better know it for the exam.